in prostate cancer. Some of this will be a little bit of review of some big studies, but I think it, where to set the stage, I'm a medical oncologist, and when I trained not that long ago, basically this is where we were in most of cancer, that we made decisions based on anatomical considerations, and we made decisions about second line therapy based solely on failure of first line therapy. It wasn't informed by anything that happened to the tumor. We just went on to second line because first line failed. And that, not surprisingly, that in lots of cancers, that made it hard to have durable outcomes. So I think a lot has changed, and I think a lot has changed in all of cancer, including GU malignancies, because of a change in sequencing technology. And this is you've probably seen a version of this. The main point of this is that sequencing technology changed over the last decade to make what was brute force simple. And so it became possible now to sequence tumors very, very cheaply, and it continues to get cheaper. And that really informed the idea of precision medicine, and the idea that you would get a tumor from a patient, analyze it molecularly, use that information to decide what therapy, and rationally then pick therapies for the patients. And there have been a couple very good examples of that. So if we take the international studies that have looked at prostate cancer genomes and look at primary prostate cancer, this was not one of the cancers that you would point as a success story in terms of genomic sequencing. What we learned was there weren't very many mutations. The mutations that occurred were in P10 and P53, and that there were a lot of copy number alterations and alterations involving this transcription factor ERG, none of which really made it easy to think about how to therapeutically intervene on these patients. However, there were a few things I think we learned in this, that the PI3 kinase pathway was important, and that hasn't yet borne out, but it may yet as time goes on. And the one thing that did come out of this that has actually turned into something clinically useful is that there were evidence of DNA damage effects, mutations, and we know that the alipridiv, a PARP inhibitor, has benefit in those patients who have those mutations. Now, almost all prostate cancer patients get treated with anti-androgen therapy, and really the question is, when they become resistant, what are the mechanisms? And so there have been a number of studies now to look at metastatic prostate cancers, and if you just look at this picture compared to the one before, what you see is there's a lot more dots all over it. And what it says is that in the resistant setting, you have more mutations. And so if we say, what are those mutations? They fall there. We don't actually know most of them, but the ones that we do know fall into the categories of AR mutations or amplifications, increased changes in DNA damage and repair, and then new mutations that affect the Wnt pathway, especially RSPON2, and cell cycle alterations. And I'll get back to that a little bit. So where we are is we now have a parts list. We know how many parts are broken. We know what they look like. But if we're thinking about how to use that information to inform patients, simply having a parts list doesn't tell you how to fix it. And that's like this. Some of you might know what all these parts are, but you really need to reverse engineer the process and be able to put this back together and understand how this tumor works inside of a patient. And so if we really want to move the precision medicine paradigm further, we really have to solve these two challenges. One is, for the majority of tumors, we don't actually know how to read the cancer genome. We have lots of variants of unknown significance, and we don't know what to do with them. And then for the ones that we believe are actually important, we don't actually have drugs. That's the second challenge. So if precision medicine is going to work for a larger number of patients, we have to solve those two challenges. And it's really the first one, and hopefully getting to the second one, that we've focused on. And so we're really good at defining what's broken in cancer cells, but we're very bad at understanding what the consequences of that. And so the tried and true paradigm in biomedical sciences, you get a lot of graduate students, you get a lot of postdocs, you give them a lot of time, and they figure things out. And that's how we've learned almost everything we know in cancer, but I'm too impatient to wait for that to happen because it takes decades. So we've really thought, well, how do we accelerate this process? And again, this goes back to knowing the genome. And so what we and others have done is taken the ability of having all knowledge of all the genes and be able to make prospective reagents that allow you to express any variant of any gene and do that systematically and test what the consequences on cancer phenotypes, or to do loss of function studies and ask the question, what happens if you eliminate the expression of particular genes? So this idea would be that if you had these tools, which we now do, you could go through and systematically figure out what are all the genes important for each cancer. All right, so I'm going to give you a quick example here in prostate cancer. A postdoc in my lab took a library of all cDNAs in the human genome, 17,000 cDNAs, and expressed them in a prostate cancer cell line that requires angiogen for proliferation, and treated those cell lines with enzalutamide, and then asked the question, which genes allow these tumor cells to survive? So a pretty simple experiment, but done at genome scale. And what the result of this experiment is that there were about 50 odd genes that allow these cells to survive. And if you look, and you can do further validation studies and show that they all allow the tumors to grow in animals as well. And what's exciting about this is if you look at these genes and say what happens to them in those metastatic prostate cancers that have been sequenced, almost all of them have been amplified. So it is highly likely that the genes that scored in the screen are amplified in metastatic prostate cancers and likely driving the metastatic phenotypes. And so the question is, what are the things that you find when you do this? And so what are the resistance mechanisms? The first is that there's reactivation of AR signaling. You get mutations, you get overexpression of AR, you get alterations in enhancers that then go back and drive the AR or allow AR to operate under lower levels of antigen. So reactivation of antigen signaling is the number one mechanism of resistance. The number two mechanism of resistance is to reactivate AR signaling. So I think the field has learned now that this is a truncal mutation for prostate cancers. When you try to ablate AR signaling, the cancer cell figures out how to turn on AR signaling. So the third mechanism of resistance is other ways to turn on AR. So I think one of the things that we've learned, we could take a lesson from breast cancer is there's never going to be enough agents to stop AR signaling. It continues to be the most validated target, and figuring out all these other mechanisms that get operated to turn on AR are still likely to be very good mechanisms for inducing longevity in our patients. Now, there were some other things. There's activation of receptor tyrosine kinases and MAP kinase pathway signaling. There's activation of Wnt signaling I mentioned before, and then disruption of the cell cycle. And so if you were to step back and say, well, how would you conceptualize this? One way to say is that all of the ones at the top all lead to AR. All of that leads to more AR signaling. And the ones down here says, if you can finally ablate AR signaling and really turn it off, what happens is a prostate cancer cell escapes by transdifferentiating into another state. And this is either the neuroendocrine phenotype or other things. And it's being seen in other cancers as well, is that at the end of the day, the only way you escape AR is to become a cell that no longer requires AR, and that is to take on the cell fate of some other type of cell. And this is going to be the new frontier about how to target these, because I think more and more of these patients are going to emerge as we have better therapies. Okay, and in the minute that remains, because I was supposed to talk about CRISPR Cas9. So the other side of this equation is to do the loss of function experiments. And I won't go through a lot of it except to say that we have a very large effort called at the Broad, where we're doing genetic screens. We've done genetic screens, whole genome loss of function screens in about 1,000 cell lines, and we're headed toward 5,000. And we've done small molecule screens in a barcoded way with Todd Golub in more than 500 cell lines, and that's also headed for a couple thousand. And the idea here is these are two types of perturbagens that would, if you put them together using cell lines as the measuring point, that we should be able to get to what are all the dependencies in cancer cell lines. And this thing that we call dependency map also takes into account all the genomic information, with the idea that not only should we develop an idea of which genes and targets are required for subsets of cancers, but what are the predictors that allow you to select for that? And so this effort is ongoing. We're working in many cancers, but certainly all of the GU malignancies are very well represented. And so the hope from this is that with this information about the function of the cancer genes and the identification of molecules, that we can fill in those two challenges and realize the promise of precision medicine. Happy to take questions.
Thanks, Bob. Niraj?
Yeah. So this was great to hear, that AR. So if you don't have the DREAM AR inhibitor at the third line setting, when AR amplification mutation is still happening, is there any way to, are there any other pathways which are allowing that AR to continue to drive the tumor signaling? So why not to target those pathways which are allowing AR to evolve and continue to drive tumor growth?
Yeah. Well, I think that one of the questions is, how do you really shut down AR? And I think the approach that's being tested clinically now is, do you move all of the anti-androgen therapies in combination up front? And does that prevent the outgrowth of resistant tumors later on? It's worked in microbiology. That's the way you avoid resistance. It's worked certainly in HIV. That's the paradigm for CML, that you put all your drugs up front and don't wait for resistance. It remains to be seen if that's actually going to play out here. I think the challenge is that if you look at the mechanisms beyond AR itself or production of more androgens or sensitivity, the real problem is that the next mechanisms are other transcription factors that get co-opted to turn on androgen signaling. And that's going to be harder to address with traditional anti-androgens.
I think, Charles Sawyer showed 15 years ago that actually the initial lesion that we really should be focusing on is not continued signaling by the AR, but AR amplification. Because AR amplification leads to all these downstream effects, and now we're going to hear about ARV7 and other things like that. What do you know about the mechanism by which AR amplification begins? Because that seems to be the most proximal lesion in the whole process.
Yeah, so I think, one of the things that is a gap in genome science right now is that when we look at genomes, we observe amplified regions, but people generally don't study the mechanisms by which those amplifications occur. Is it a DFHR type mechanism of amplifying or making mini chromosomes? It's not clear. I think with androgen, what is clear, if you look at the structure of the amplicons, is that not only are you amplifying AR, but you're amplifying enhancers that are driving more AR signaling.
Right. So as a corollary to that question, the data coming out of Hopkins looking at giving high-dose testosterone to patients with prostate cancer and showing responses, what is different about that signature? Have you looked at that particular question? Because clearly, you don't want to do harm to patients by supplementing back testosterone. Is it because you're downregulating the androgen receptor expression by giving T back? Has there been a mechanism that's been postulated?
Yeah, I think it's a good question. I think part of our challenge to answer some of these questions is that we can only answer them in people. Because one of the real limitations of the field continues to be the bona fide models, experimental models of prostate cancer. We have very, very few, if any, mouse models that really look like human prostate cancer. And as you know, there's still only 4 or 5 cell lines out there. So that's a really important question to ask and get answered. But I think we don't have the materials to do it. And the only way to do it is clinical studies, which obviously take a long time.
So, I'll point out, a series of studies from Hopkins and a little anecdote or two from us. But the high-dose testosterone really can shut it down. So we were monitoring circulating free DNA and be able to turn a patient who had almost a 17-fold amplification on cfDNA down back to normal with high-dose T. We sensitized him to the use of an abiraterone, or even enzalutamide a little bit later, But it was really quite interesting. We went back and forth on this patient, and we could follow the molecular alterations in serum. Really cool and real time in patients that you can do this. So maybe we're going to get there faster than you think. It's
Yeah.
possible.
Well, I think that, the other question here is the pharmacology issue. What is the right schedule and order to do all of these manipulations?
Yep. Certainly don't know the answer to that. And I'm sorry, another question.
Bill, have you tried any of this in organoids or in conventionally reprogrammed cultures from patients? You mentioned the limitations of the models in terms of making functional predictions in patients, given the heterogeneity of disease that we see clinically.
Yeah, so we have a big effort with Matthew Ellis and others to try to adapt the tools that we have to do them in organoids. We can't do genome scale studies, but we can do smaller scale studies, and that's where we're started. So I don't have any data yet, but we're trying.
All right, we're going to go ahead and